Hey there, and welcome to another MP3 tutor session. This is Eric Simon, and I'll guide you through today's topic, proteins. In your text and in other MP3 sessions, you've been exposed to one of the major themes of biology, the relationship between structure and function. In biology, we say that form follows function. That is, the structure of an object reflects the job done. For example, the form of a chair must reflect its function. Chairs come in many different shapes and sizes, but they all must have a flat part to sit on and a stable part to meet the floor. Throughout multiple levels of biological organization, exploring the theme of structure and function can help you to develop a deeper understanding of how life works. In this MP3 tutor session, we will explore how proteins are put together and relate that structure to a few specific examples of protein function. Sound good? Let's get started. When I hear the word protein, the first thing that usually pops in my mind is food. Meat, dairy products, eggs, that sort of thing. Of course, I think about food a lot. However, even when my appetite is saying salad, I always make sure that my salad has ample protein in the form of nuts, seeds, and beans. Proteins in food provide the raw material to build the bodily proteins essential for your good health. Proteins have the most diverse and complex architecture of any of the biological macromolecules. You have tens of thousands of different proteins in your body that have diverse structures and perform diverse jobs. Proteins are the worker bees of your body. They take part in nearly every activity of an organism. Proteins play a role in structural support, as in cartilage, transport, such as hemoglobin, hormones, like insulin, immunity, your antibodies, movement, as with muscle proteins, and as enzymes that keep thousands of different chemical reactions running inside all cells. In each of these examples, the shape or structure of a protein is the key to understanding how it works. How can the shape of a protein be so important? Let's try an analogy. Pliers are one of the most commonly used tools for extending the power and grip of your hand. Most people have used pliers to accomplish a specific task, such as cracking a nut, removing a bolt, bending wire, cutting wire, so on. In each case, the movable jaws of the pliers are shaped for specific tasks. Using them for a different duty sometimes leads to frustration. For example, you can't remove a bolt with a pair of needle-nose pliers. Shape is important to a tool's function, and the same is true for proteins. Each of the thousands of different kinds of proteins in your body has its own unique shape. Describing so many different proteins with so many different shapes sounds like a daunting task. Luckily, it's not as difficult as it sounds. Let's explore how proteins are put together. When describing the structure of proteins, biologists think about 4 distinct levels of protein structure. These 4 levels are known as primary, secondary, tertiary, and quaternary. I love that last word, quaternary. I actually had to look it up in the dictionary. Turns out it can also be pronounced quaternary. Anyway, I always have trouble pronouncing these words, so sometimes I simply refer to these levels as the first, second, third, or fourth level of protein structure. The first, or primary level of protein structure, is simply the linear sequence of amino acids that are strung together to make the protein chain. Every protein has a unique amino acid sequence. Just as you can build thousands of words using just 26 letters of the English alphabet, a huge variety of proteins can be constructed from 20 different amino acids. Just like English letters have different properties, such as consonants versus vowels, so do amino acids. The properties of an individual amino acid lay in its side chain that branches off the carbon backbone. The side chain can also be called the R group or radical group. Let me use another analogy. Different railroad cars, such as tanker cars, flatbed cars, or refrigerator cars, can be built on a standard railroad car chassis so that the cars can be easily linked together to form a long train. Likewise, a common carbon backbone is found in all amino acids. This common backbone permits amino acids to be linked together in long chains. Unlike a train, however, the chain of amino acids folds and twists to form a complex three-dimensional shape. The shape eventually taken by a chain of amino acids is directly tied to the side groups that protrude from the amino acid chain. If even one amino acid is out of order, it can make the entire protein non-functioning. However, let me point out that some amino acids can be substituted for others without altering the function of a protein. Let's go back to the word analogy for a second. If you change just one letter in some words, you can completely change the meaning. For example, changing one letter in the word ship can produce a completely different word like shop. Very different meanings, right? Other words, however, are not as susceptible to single changes. If you change a letter around, they may be misspelled, but their meaning may not be changed. For example, if you switch around the E and the I in the word protein, you'd have no trouble understanding the meaning of the word. If the amino acid sequence of a protein is changed, most changes result in defective proteins, but some do not. To recap, the primary structure of a protein is simply the list of amino acids in their proper order. To understand the secondary level of protein structure, imagine that you are an alien seeing a room full of human beings for the first time. The first thing you might notice is that every person has a unique shape, but you would quickly start to notice similarities in the shapes. Every person, for example, has a rectangular body with a spherical blob at the top and 4 long limbs sticking out. If you look at many proteins, you will similarly notice certain common structural elements. The two most common are called an alpha helix and a beta sheet. An alpha helix is a region of a protein that is twisted into a tight spiral. The coils of the helix are held together by hydrogen bonds between nearby amino acids. Other regions of a protein fold up like a paper fan. This type of structure is referred to as a beta sheet. The alpha helices and the beta sheets together comprise the secondary structure of a protein. A given protein might have 3 alpha helices and 4 beta sheets, while another has 8 and 2, and so on. Most proteins have at least some of each. Once all the helices and sheets are coiled, folded, and locked into place, the polypeptide takes on a distinct 3-dimensional shape. This is the third or tertiary level of protein structure, the overall conformation or 3-dimensional shape of a protein. To think about protein shapes, imagine that I have a big barrel of clay and I ask everyone in the class to grab a lump and put it on their desk. At first glance, all the lumps look similar. They're all blobby lumps of clay. But closer observation reveals that every lump of clay is unique. Some are taller or fatter, some have bumps and grooves that others lack. Similarly, while most proteins look basically like roundish blobs, each one has small differences in their structure. And these differences in shape are vital to the job a protein performs. That is, form follows function. Change a protein's shape in the slightest, by say cutting off a lump, and the protein may not be able to perform its job within the cell. The fourth level of protein structure describes interactions among different polypeptides. Some complex proteins are composed of more than one polypeptide chain that bond together to form a sort of superprotein. For example, the blood protein hemoglobin is composed of 4 different polypeptide chains that interlock. Another example is the molecular machine known as DNA polymerase, the enzyme that builds new DNA molecules during DNA replication. DNA polymerase is actually a large complex made up of a number of different polypeptide chains joined together. Not all proteins have quaternary structure, but the big ones usually do. As a summary, let's review the 4 levels of protein structure. The first level is the sequence of amino acids. The second level is the formation of alpha helices and beta sheets. The third level is the folding of a polypeptide into its final 3-dimensional shape. The fourth level is the joining of more than one chain to form a larger complex. I hope this gives you a leg up on this topic. If you need more help, please refer to your book, CD-ROM, or course website. Good luck with your studies.